Hello, and welcome to the American Floral Endowment's three-part webinar series for Botrytis Research. Throughout this series, we will hear the latest findings from AFE's Thrips Botrytis Research Fund. AFE is the national nonprofit organization that funds scientific research to identify and solve challenges within the floriculture industry. In 2021, AFE is celebrating 60 years of providing for the future of floral. In 2017, after listening to important industry feedback, AFE established a special research fund to aggressively address the control and management of Thrips Botrytis. AFE's goal of reaching 1.5 million in pledges was met in 2019, with contributions from 24 industry leaders and organizations to support new and innovative research to address these challenges. With those funds, AFE has been able to support 8 multi-year research projects to reduce losses and produce higher quality flowers and plants. The American Floral Endowment and all researchers would like to thank all of the organizations who have made contributions in support of this important initiative. Today's speaker is Dr. Jim Faust, Associate Professor of Floriculture Physiology at Clemson University.
Today we're going to do part 3, and that is looking at strategic management principles for gray mold management. And before I get into that, I'll kind of do a very quick overview of what we have covered thus far. In the part 1, we talked about reducing disease pressure in the greenhouse through crop sanitation practices, disease monitoring strategies, and creating a drier environment so that we're reducing the spore population. Then in part 2, we've started out talking about maintaining fungicide efficacy. In other words, minimizing fungicide resistance, and doing so by understanding the roles of single and multi-site fungicides, identifying fungicide resistance development, and evaluating fungicide resistance or efficacy, in-house. So we talked about how you could develop a lab to do some of this efficacy testing yourself. And then really as a continuation of part 2, the topic of part 2, we are going to delve into part 3 today. And that is looking at alternative disease control products, basically beyond fungicide application. And we're looking at use of these products during periods of low risk of infection. Essentially, we're reserving the best products, which are the single-site fungicides, for the most important times of the year. But then when we have lower risk, or because of the environment, or if we have an off season, so it's not as critical to have 100% control, then we can use some of these other products that are not going to be perhaps as effective as single-site fungicides, but they will have a degree of efficacy and will allow you to create fallow periods where you're not using the single-site products, so that when you go to use them, they will be effective. So we'll be and our options are multi-site fungicides, which we have already talked about, but we'll review briefly. And we'll focus mainly today on the use of calcium, the use of biological control agents, and we'll mention how ethylene is also a part of the botrytis management package or strategies. So just to review very quickly from last week, the multi-site fungicides, these are broad spectrum products. They are preventatives or protectants. They are not curatives. They don't have a high level of efficacy like the single site, but they have a moderate level of efficacy. They do not penetrate tissues. They stay on the surfaces. They kill spores on contact and prevent spore germination. But once the spore is germinated, they are not effective. So they really need to be a protectant on the surfaces before the spore lands. The application technique requires good coverage, essentially small, well-distributed droplets. And then we have with these a low risk of resistance development. So that's the multi-site story. And again, we talked about more of that last week, or in more detail. This week we'll focus on calcium. Calcium reinforces and stabilizes the structure of the cell. And so this is our little illustration where you have 2 plant cells, and then the intercellular space is calcium, when it's available in good quantities, forms this matrix with pectin to create a kind of a glue of sorts to provide stronger structural integrity to plant tissues, and therefore making cells more difficult for Botrytis to penetrate into. We showed you one of these pictures in part 1, I believe. On the right is our example of low calcium content. So these little black things are just our representation of calcium concentration. And so this is a low concentration scenario. We have spore germinates, germ tube forms, you have penetration, mycelium penetrates the cells, steals the goodies, kills the cells in the process. And whereas if we have a higher calcium content, what we see, the spore will still germinate and tries to begin to penetrate the tissue, but fails to do that very effectively. So we limit the ability of the spore to infiltrate that tissue and then to eventually sporulate and create a huge population of spores in your facility. So this is what calcium does. And we know of no known resistance to calcium, unlike fungicides. So we'll show you a few data slides today. The first one, it's such a dramatic piece of information that it's useful to see the actual data. So this shows you calcium content in tissue, and if you've all sent tissue analysis to the labs, and usually it's the most newly unfolded, fully mature leaf, and that tissue is typical of this data here, where you'll have at least 1%, upwards of 2% calcium in that tissue. The sepals, the green calyx below of this, in this example, this is with roses, so it's the green sepals below the petals. You also have high concentration of calcium. But when you look at the petals of the rose flower here, oops, you see a really low level of calcium. It's less than 10%, down near 5% of the tissue content, the leaf tissue content. The stem is also relatively low, a bit higher than the petals, but it's no wonder that we see an association between calcium and Botrytis infection, because these tissues that are really low in calcium are the ones that are really susceptible to Botrytis. We've collected a lot of roses, shipped internationally, and we put them in human chambers and see where Botrytis infections have occurred. And in all of these roses, what we see is almost 75% of the infected tissue that arrives on cut flower roses is on the petal tissue. There's a little bit in the stem and the sepals, but you see these distribution looks very similar to the calcium concentration in those tissues. So then the question is, how do we maximize calcium concentration in the tissues? That's what we're trying to do. And what you'll see, I don't have this in the slide, but I just thought I should mention it. If you look at hydroponic growers, they use much higher calcium levels in their solutions than ornamental growers. And I think they have found that it's so essential to have adequate calcium in crops like lettuce to prevent leaf tip burn, or in tomatoes to prevent bract edge burn. And also you get Botrytis on the stems on tomatoes pretty easily. And so typically in these hydroponic solutions, the calcium content is over 100%, upwards of 200%. I saw one California grower show me once his tomato recipe, and it was 300 parts per million calcium. And in ornamentals, we're often applying just 50 parts per million, 35 parts per million, fairly low numbers. Now we do have some calcium in the growing media that hydroponics don't have. So gypsum is a source of calcium, and the lime can be a source of calcium, but we tend to apply a good bit less than vegetable growers. And I just think it's because we've gone to these one bag fertilizer solutions, and to get really high concentrations of calcium in your fertilizer solution, you often have to have 2 tanks, because you can't mix the calcium at a high rate in a one tank system. Anyway, I'm regressing. The three ways to maximize calcium concentration. The first one is fertigation. And so this is an effective technique only in some situations. and it's effective where we're delivering calcium to transpiring tissues such as leaves, because over the life of that leaf, it has a big opportunity to accumulate calcium. Whereas when we have rapidly developing tissues that are low transpiration tissues, like flower petals, petals don't transpire a lot of water and they develop very rapidly. So there's not a big window of opportunity for calcium to move, into those tissues. So the other thing is with calcium is that the environmental conditions impact the amount of transpiration and thus the amount of calcium transported into these tissues. And I will illustrate that with this little diagram. When we talk about plant nutrition, there are two ways that nutrients are taken up. There's active uptake and passive uptake. Most plant nutrients, nitrogen, phosphorus, potassium, magnesium, sulfur, are taken up actively, meaning the plant will spend energy at the root interface to uptake those nutrients, and as a result, the plant can have a higher concentration of the nutrients inside the root than outside the root. And then as those nutrients are taken up, the nutrients in the soil will diffuse toward the rhizosphere because they'll be moving from an area of high concentration to an area of low concentration. However, calcium is taken up by mass flow, meaning it comes along for the ride with water that's taken into the root that is going up to the plant to supply the plant with water and to be transpired. So the amount of uptake depends on the amount of uptake of calcium depends on the amount of mass flow. So if we have a scenario here where we're showing a small arrow for mass flow, there's a little bit of water going into here because it's having low transpiration in the environment. We have low mass flow and low calcium uptake. We get low transpiration when we have cool, cloudy, humid weather. Active uptake is not influenced, right? Because the plant's actually spending energy to take up those nutrients. The plant is not spending energy for mass flow. It's just simply moving along as water leaves the leaf. It sucks up water through the stems and through the roots. And so if we don't have a lot of transpiration, we have low mass flow, low calcium uptake. In contrast, if we have a condition, for example here, high transpiration, sunny, warm, dry weather, we have high mass flow, high calcium uptake. And so with this high mass flow, a lot of calcium moves into that tissue. So calcium is unique compared to the other nutrients in how we manage it. And as a result, in greenhouses, which can be humid, cloudy or cool spring weather conditions, we often are not getting adequate calcium move into these rapidly expanding petals of flower tissue. If we do a calcium test or tissue analysis, we'll find adequate calcium in the leaves, but inadequate in the flowers. The second way we can get calcium into plant tissue is through spray or foliar applications. In this situation, we are directly delivering calcium to this low transpiring tissue or non-transpiring tissue. For this to be effective, the spray has to come in contact with the tissue to be protected because calcium is not readily transported through the plant. You spray it on the leaf, it's not going to move to other leaves. You just spray it on one leaf. You spray it on one petal, it's not going to move to other petals. So sprays must come in contact with the tissue. Our first work with petunia was really successful, and we really realized later, petunia is typically one big petal. And as the bud is developing, we're spraying the backside of the bud, which is the backside of the petal. So as that flower is developing, we can get calcium into the petal fairly well. And so these foliar applications work really well on single flowering species. We get a more complicated situation on double flowering species, which we have a lot of in ornamentals. So this would be something like a rose, where the outer petals it's easy to spray calcium on them, but the calcium is not going to move from that petal down and then back up to the next petal. It's not going to move across those petals. So the calcium is limited to that outer petal in a similar way that fungicides are limited. We spray the backs of the petals as that flower is developing, and when that flower actually opens up, the inner petals are not protected by the fungicide. They're not protected by calcium either. And so this makes it an additional challenge of using calcium for this double flowering form. So what it means, we have to somehow get the calcium in contact with those inner petals, and that means spraying late in the crop cycle. And we'll go back to some work that Katie Bennett did in my lab a few years ago that I think is also interesting. What it shows is how calcium does move within a petal somewhat, but doesn't entirely. So for example, we sprayed this flower with 1,500 parts per million calcium. And this one, we covered up the left side of the flower. So we just sprayed the right side of the flower with that same calcium spray. And then this one was not sprayed. And then we inoculate them with Botrytis spores. And of course, the one without calcium deteriorates quickly. The side of the flower that does not get sprayed has some infection, but it's not nearly as bad as this over here. So our analysis of this, or interpretation of this, is that we are getting some movement across that tissue. So you don't have to have perfect spray application, but on a single flowering form. And then of course you get nice protection on the with the calcium on the where we sprayed the entire flower. Katie also showed that really this demonstrates the effectiveness of calcium. So we have 6 petunia flowers that are our control. We don't spray any botrytis on them. You will see botrytis infection. These are put in a humid chamber, so spores that are on the tissue will germinate and grow. And so you see some infection, and that's just from spores that happen to be in our greenhouses. If we inoculate the plants with a suspension of spores, a solution of spores, we wipe those flowers out pretty quickly. If we pre-treat the flowers with fungicide and then inoculate them, in this example, the fungicide performed very poorly. And then we did some fungicide resistance analysis and realized that this fungicide which we were using, which at that time it was palladium, which is a very good single site fungicide, but these plants obviously had resistance, and we tested and proved that these Botrytis spores that we were spraying as part of this experiment were resistant to palladium, or switch is another name for it. And so then we did calcium at a low rate, and this is a rate that historically was fairly common when people would spray calcium on for things like bract edge burn on poinsettias. We did 800, thought we'd be a little bit crazy, and saw a benefit, but still not great. And then we did 1,200 and really had some very clean plants that were effectively better than our control. So these guys were not inoculated on the upper left. These were inoculated with thousands of spores, and they were resistant because they had been treated weekly. I believe it's for 4 weeks prior to the spores being applied. So calcium can work really well if we get it in the right place at the right time. And we can never get this level of calcium in the tissue through fertigation. You could fertilize with as high a concentration as you can mix up, and you would not get enough calcium in that flower petal tissue to get this sort of result. All right, so the third way that we can get calcium then is through a post-harvest treatment. Either in cut flowers we can dip, or provide a heavy shower, or we could do this on bedding plants, for example, too, in that we have to get enough solution that it goes down into these double flowers, or dunking the flowers works really well. So we're really targeting the petals that have not yet been treated in the greenhouse because they've just opened up in the last 3 or 4 days before shipping, for example. It's important to use a wetting agent because when you have these layers of petals, the water solution doesn't move down into those and actually coat those petals well unless there is a surfactant adjuvant of some sort. So this is an effective treatment. So this is our third way. Just to show you how effective it can be, this is an experiment with roses where we took roses that were packaged, processed, and ready to go into the box for shipping. And we dunked them in a solution of calcium chloride. And these are the results we got. So these are the left-hand column here is a hydrogen peroxide solution that we dipped in because that's a typical thing done commercially. And then we have 0, 1,000, and 2,000 parts per million calcium dips. And you can see the botrytis infections occurring over time and this growing and growing and being pretty severe at 12 days, better at 1,000 and really quite clean. You're starting to see a little bit of infection here, but again, 12 days is pretty impressive at a higher rate than you may have ever considered using calcium before. But we never saw any phytotoxicity on these with 2,000. I would say on some sprays in the greenhouse, I've heard of growers having some phyto using over 1,000 parts per million. But when we've seen phyto on petunias, which is the crop we've worked on the most, it's fairly small and fairly short term. It's not a devastating phyto, but if you look, you might see some spotting. So that's the calcium story. For the botrytis, the biological control agents, this is our newest area of research that we're doing through the American Floral Endowment, and also including systemic acquired resistance inducers. So these are compounds that we apply to plants that induce natural resistance to botrytis in those tissues. So the plant creates compounds that help the plant fend off botrytis. So these are examples of the products that we will be working on. We have started work on some of them already, and some of these are commercial products, some are still experimental. And the main species that we're working with are Gerber, Rose, Petunia, and Poinsettia. So we'll show you a few data slides. This is all preliminary, but I want you to see what direction we're going in and to see how this might fit, and how you might want to initiate some trials on your own with some of these products because of their potential value. So where we've started was with what we're calling in vitro assays. So not working in a plant system, but simply taking our biological control agent on a petri dish. We make some spots on a gel, so there's a food source for it to grow. So there's our in this case, I think we have bacillus, and then we put some spores of botrytis in the center and watch them grow. And what you see is the botrytis is growing, but not coming in contact with the bacillus, and it's kind of growing around it in a way, kind of avoiding it. If it grows over top of the biocontrol agent, then that agent's obviously not doing anything to repel its growth. But in this case, this has indicated that, okay, this is a product that has some capacity to be used. And so basically what we're, I'm sorry, so there's our botrytis in the middle. So we're screening these different biocontrol agents with botrytis spores that we've collected from different species, and just to see which ones might actually look good for future studies. This is some data just to show you, this is reduction in botrytis growth for about 8 different products here, 9. And these 3 are trichoderma products. And you can see we're looking at 60% reductions in growth. So that's pretty good. The bacteria products that we're using are a little lower, but still effective. And this is kind of what we're looking at with BCAs, is that you're not looking at something that's as effective as a single site fungicide, but it does work if used appropriately. And so if we could use these products again during periods of low risk, or periods that are non-peak seasons, then these products will allow our single site fungicides to last for more years and be more effective when we use them. So now what we're doing is in vivo treatment. So we're actually using intact plants. These are the first 6 products that we're looking at, and we're looking at them as sprays and as drenches. They may not necessarily be labeled for both, but we're trying them as both. And just again, this should just show you some initial results that I think are hopeful. We show disease severity over here. So 0 is no disease and 8 is bad. And then we have a bunch of treatments, and we have D's are drenches, S's are spray. So water, drench, spray, you can see is the same as the non-sprayed product. Then we have cease drench, spray, triathlon, and so on. And then on this side, we have where we have not inoculated in these two treatments. So there's not spores applied. And so we don't have disease severity. But you'll see this one, actually the best, this one and Cease. So this is On Guard and Cease are our two best products. On Guard is an organic form of calcium. So it's the same mode of action as the calcium sprays. It just is a different formulation. It's calcium bonded to a plant protein, soy protein. So it's, I believe it's Omni labeled, Omri. And so that looked really good. Cease as a spray looked really good. You actually see that On Guard as a drench was effective because again, you're getting calcium into the plant and then in an alternative way as a fertigation treatment. What's interesting, we have Captan, which is our multi-site fungicide, and you can see it's moderate. You can see the calcium, On Guard calcium was better than the Captan. And we see a number of these other treatments were equal to the Captan. Again, a moderate efficacy, not outstanding, but it's not nothing. It's not snake oil. It does work. And so it has some, some real potential as again, to give us an alternative. It's probably not going to replace single site fungicides unless you have a crop that is not highly sensitive or if you're in a low risk situation, you might not need the single site fungicides at all. So anyway, I just want to point out those. And then our moderate treatments here, again, are ones that were similar to the Captan spray. Captan drench did not do anything, but the sprays were similar to On Guard drench, root shield drench, Guard spray, triathlon spray. Okay. The last point I wanted to make is that John Dole and Ben Bergman and NC State have done some nice work in recent years showing that within roses, we get a degree of variability and susceptibility to botrytis. And there's actually a correlation between the varieties that are more ethylene sensitive and varieties that are botrytis sensitive. So, ethylene being a signal of plant stress and aging, and it appears that if ethylene sensitivity is linked or at least correlated with botrytis sensitivity. So if we can apply ethylene control management techniques to control the ethylene the plant is perceiving, we think the botrytis can also be managed or reduced in post-harvest environments. So this would be using things like ethyl block or STS in a post-harvest situation. We think will provide some assistance in managing botrytis in the post-harvest environment. So that's promising also. So we'll leave you with this last slide. And this is basically our decision-making strategy approach that I've now mentioned a few times, but we'll summarize here. We've kind of created our 4 potential scenarios. You have a peak or high season or a low or off season. And then you have a high risk because of the environment being humid, a lot of condensation, cool, cloudy conditions, or a period of low risk where it's drier, sunnier, or warmer. And the idea is high risk, peak season, we need to be using our best products. But if we are going toward the low risk, lower peak season or less critical time of year to have absolutely good control, we should not be using these single site products. We should be relying more on these alternative approaches, BCAs, SARs, calcium, ethylene. We don't think these guys over here are eliminating the need for good fungicides. But if we use them when we want to use the good fungicides, we want to know that they're going to be effective. So relying on the guys on the right side more often will be help us manage botrytis better. So we focus on single site fungicides during periods of high risk. Multi-site fungicides do need to be rotated if we're applying them regularly. Although the risk of resistance is low, it's not zero, and so you still do rotation. The other thing to note with the biological controls is the way they work. They're not they often need to colonize space on the plant in order to keep the botrytis from growing in that same space. So they're competing. They also can be releasing toxins to inhibit the growth of those of botrytis. So what's important is that usually one application of any of these products is not provide a long-term solution. What you're looking at is establishing a population of beneficials or continuing to apply, say, calcium into newly emerging tissue, so you're protecting it. And so it really requires regular application. Also, I'm a little concerned that, I would think that the biological controls are probably going to be much better. I don't have data for this yet, but I think they're probably going to be much better in the canopy, where the leaves are there for a long period of time, as opposed to fast emerging, fast expanding flowers. It's going to be harder to establish good populations of biologicals on a flower petal before a botrytis spore lands on that petal. So it could be that the single site products are what we really need to be focused on when we're getting into that last week or two before selling a flower, and during the vegetative phase, during the leading up phase, we're using the biologicals to keep the spore population low. So again, foliar applications of calcium in production and in the post-harvest environment can work. And again, multi-site fungicides as well as the biologicals and calcium really require good, thorough coverage. And then lastly, we should consider and should further look at ethylene treatments as having potential to minimize or reduce botrytis infection.